Hi, my name is Betsy Holmes, and I'll be presenting to you on the DNA sequencing method, Dideoxyribonucleotide chain termination sequencing. So what is DNA sequencing? DNA sequencing is a laboratory technique that uses the principle of complementary base pairing to determine the exact sequence of nucleotides in a DNA molecule. Complementary base pairing is the phenomenon in which guanine always bonds with cytosine, and adenine always bonds with thymine, and the sequence of nucleotides in a DNA molecule carries the necessary information a cell needs to assemble protein and RNA molecules. So what is dideoxyribonucleotide chain termination sequencing? Also referred to as dideoxy sequencing, it is the first automated DNA sequencing procedure working with about a maximum of 900 base pairs at a time. This procedure was developed by the British biochemist Frederick Sanger, who won the Nobel Prize in Chemistry twice. How does dideoxy sequencing work? In simpler terms, one strand of a DNA fragment is used as a template for the synthesis of a nested set of complementary fragments. These resulting fragments are then assembled into a continuous sequence of larger portions of DNA and eventually chromosomes. In more complex terms, dideoxy sequencing can be split into three steps. The first step is a chain termination polymerase chain reaction, or chain termination PCR. A typical PCR reaction has the usual steps of denaturation of the double helix, annealing to initiate polymerization, and extension to synthesize the new strands of DNA. However, in chain termination PCR, chain terminating modified dideoxyribonucleotides, shortened to DDNTPs, are mixed into the reaction. These DDNTPs lack a hydroxyl group, which is needed for phosphodiester bond formation. This results in an increase in the extension step when DNA polymerase incorporates a DDNTP at random. Step 1 of dideoxy sequencing results in millions of oligonucleotide copies of the targeted DNA sequence. The second step is size separation by gel electrophoresis. In this step, oligonucleotides from the first step are separated through a standard gel electrophoresis process. To perform this, the sample is loaded into the gel matrix and the electrical current is applied. DNA has a negative charge, so when the current is applied, the fragments will be pulled towards the positive charge on the opposite end of the gel. Because DNA has the same charge per unit of mass, the speed at which oligonucleotides move is indicative of their size. Step 2 of dideoxy sequencing results in arrangement of oligonucleotides from smallest to largest, reading from the bottom to the top of the gel matrix. The third and final step of dideoxy sequencing is gel analysis and determination of DNA sequence. In this step, a computer reads each band of oligonucleotides in the gel to determine the sequence of the input DNA. The bands are analyzed with the knowledge that DNA polymerase only synthesizes in the 5 to 3 direction starting at a given primer, so each terminal DDNTP will correspond to a specific nucleotide in the original sequence. This results in the determination of the 5 to 3 sequence of the original DNA strand. How is dideoxy sequencing used? As the first automated sequencing method, it was used in the Human Genome Project. This project sought to sequence the human genome, and 20 large sequencing centers in 6 countries were involved in it. With no other automated sequencing methods at the time, Sanger's dideoxy sequencing method was the primary method used in the beginning. However, there are about 3 billion base pairs in a haploid set of human chromosomes, and the dideoxy sequencing method worked on a rather small scale. This project served as an impetus for the development of technology for faster and larger scale sequencing methods, which eventually led to the next generation sequencing method. Although there are sequencing methods that can handle larger portions of DNA in shorter amounts of time, the dideoxy sequencing method is still widely used today for the sequencing of individual pieces of DNA. Two instances where dideoxy sequencing is still used is in DNA cloning and polymerase chain reactions, as these are smaller scale, detailed processes. DNA cloning is the process of making multiple identical copies of a particular piece of DNA, and a polymerase chain reaction is a common laboratory technique used to make many copies of a particular region of DNA. Thank you for watching, and here are my resources.